So, good afternoon, everyone. And on behalf of IRR program and experimental surgery graduate program, I would like to thank and introduce Dr. Megan Kiryan from Michigan Medicine. And first of all, many thanks for accepting the invitation and enlightening us with your amazing research work. So Dr. Killian is a tenure track assistant professor at the University of Michigan in Orthopaedic Research Laboratory and the Department of Orthopedic Surgery. Her research portfolio includes development of preclinical translational models of studying development injury and healing of tendon-bone interface. Her recent work is focused on the role of fibroblast growth factor, FGFs, mechanical loading and metabolic disorders, specifically mitochondrial dysfunction, energy metabolism, and hypoxia, which personally got me very excited because I kind of want to work or I'm working on that. So Dr. Killian has received her Master of Science from Montana State University and training in meniscus mechanobiology from Michigan Technological University, and did her postdoctoral training at Washington University School of Medicine, where she was supported by an F-32 grant, and she worked on rotator cuff degeneration and healing and the postnatal growth of the bone tendon interface. Thereon, she moved over to University of Delaware to set up her first lab as her first independent investigator. And then in 2020, she moved to University of Michigan. She has been bestowed with many, many awards, like recognition from Orthopaedic Research Society, Innovation and Career Awards from Biomedical Engineering Society, the Ruth Kirstein National Research Award from NIAMS, the Early Career Award from Journal of Orthopedic Surgery, and Career Award from NSF. So without further extension and abuse, I will just invite Dr. Killian to show the amazing work that she does in her laboratory. Thank you, Megan.
Thank you so much for that very kind introduction. And I should preface it with, I'm not doing a whole lot of the work myself these days in the laboratory. It's being led by very talented students and technicians in the lab. But thank you. I appreciate that. So some of you I have recently found or have been following for a while on Twitter. So if you're on Twitter, it's usually a good way of getting a hold of me. I have a personal account as well as a lab account. We're also on Instagram, but we don't tweet as much, but feel free to send me a message anytime to have a chat. So I don't have any financial disclosures or conflicts related to this presentation. And as I walk through the presentation, if at any time you would like me to clarify something or would like to ask a question, feel free to raise your hand or use the chat. I'll keep that open while I'm presenting to monitor as we go. And then I'm also going to share a Google form. This form is better filled out at the end, but this is just for me personally to get feedback from you all on how I'm presenting and a little bit of self-interest here for collecting data for improving my talks as I give them, and also using these as data for future promotion and tenure dossiers when that time arises. So I'm going to drop the link here and I'll add it at the end. I'll try to remember again. And then I just like to, if you're going to have your little video of me speaking in your screen, if you don't use the full screen, it's not an issue, but if you do, you can put your video here. I'll try to keep data out of that region for the rest of the talk. Okay, so those are all the disclosures. One more I have is that the World Congress of Biomechanics is approaching next summer. We'll be in Taipei. And abstract deadlines are next week. So I'm chairing a session on embryonic and developmental mechanobiology in the mechanobiology session. And that's with Lance Davidson at UPitt. And we're looking for great abstracts. So if you're doing any work in biomechanics or embryonic development or mechanobiology, would love to read them and invite you for talks there. Okay. So the work that I do in the lab, or the work that the group does, is focused on the interface between tendon and bone, or we refer to it as the enthesis, and it's often called the enthesis in the literature. And this tissue is a fibrocartilage interface that dissipates localized strains that arise between tendon, which is a very soft and compliant structure, and bone, which is hard and mineralized. And this interface, or this enthesis, is a graded mineralized tissue that includes that transitions from bone to a calcified fibrocartilage into an uncalcified fibrocartilage, where the tide mark separates these two, and then into tendon. And this structured tissue has cellular morphology that changes in a gradient fashion from tendon, which is aligned fibroblasts, into the enthesis, which become more fibrochondrocyte-like. There's some chondrocyte-like cells and fibroblast-like cells intermixed, and these cells here at this interface during growth and development express both chondrogenic and tenogenic markers. And then these cells interface with transition into bone and more osteoblast or osteocyte-like cells. This tissue is graded cellularly in morphology, but also structurally at the matrix level, where you have aligned collagen type 1 in the tendon, which interfaces into the fibrocartilage, which is primarily collagen type 2. And this enthesis tissue has a structured, the fibrils in this interface kind of fan out and interdigitate with bone. These fibers get skinnier and change their orientation as they interface with bone. And also in some of these regions start to deposit mineral during the growth phase. And that's kind of shown here in C from this paper from Nature Materials a few years ago that shows different changes in collagen type, as well as the size and orientation of these fibers as they interdigitate. The blood supply in the enthesis is pretty low, as is in tendon. And I show here this mineral gradient where tendon is unmineralized and becomes more mineralized as you move towards the bone. So the primary focus of the work in the lab is to understand how this tendon-bone interface develops and also adapts to mechanical loading, such that we can develop new therapeutics and engineered materials to help guide regeneration of this tissue when it's injured. And the enthesis is actually quite tough. However, degenerative changes to the enthesis happen with age and with overuse, and this is commonly seen in the rotator cuff. So the tendon to bone interface in the healthy rotator cuff tendon, shown here, has that nice graded structure, and in injury, when it repairs, it heals via scar, fibrotic scar. It has increased vasculature, disorganized collagen fibrils, or collagen organization, and just increased cellularity. And so we're working with groups in chemical engineering and material science and biomedical engineering to translate materials that promote regenerative regeneration of the enthesis following injury. And a lot of the work that goes into designing these materials is driven by what we learn about how the tendon bone develops and adapts to mechanical loading. So mechanical forces are essential for development of the musculoskeletal system, especially bone and the tendon-bone interface. We know that loss of mechanical loading due to loss of muscle contractions can lead to reduced chondrocyte proliferation. This can impair bone elongation because the growth plates don't expand as much. Additionally, in the growth plate, column formation of chondrocytes can be inhibited, and the organization of the resting chondrocytes is impaired. And this can also affect elongation of the bone. Absence of mechanical loading and contractions from skeletal muscle can also impair the growth of eminences where tendon attaches to bone. And while the eminences still initiate, they form, they don't continue to grow, and they are not maintained. Additionally, there is no differential growth in long bones in the absence of mechanical loads. These in the absence of mechanical load, you don't get curvature of bones, you just get like cylindrical, circular, circumferential shapes. And lastly, we often see not all joints, but many joints will fail to cavitate in the absence of muscle contractions. So this loading of the tendon by muscle contractions as it interfaces with bone really drives how bone develops and responds to mechanical loading. So there are several injuries, like I mentioned, rotator cuff injuries associated with tendon loading during the growth period. That's more of an aging related disorder. During growth, there are also injuries associated with it, typically apophysitis, where you see this expansion of the growth plate by pulling on the tendon near that interface. And that appears in cases like Sever disease about the heel, and Osgood-Schlatter syndrome in the tibial apophysis, where you get these pulling of the patellar tendon and microfractures happening at that interface to the bone. And this can lead to a lot of pain and disuse in kids, causing them to reduce their activity and feel a little bit better. So we want to understand how these changes happen, how we might be able to tailor loading regimes for kids so that they don't experience these types of loading induced injuries, but also can remain active and grow in their skeletons. So as I mentioned, the enthesis is a functionally graded tissue, and this tissue reduces stress and strains, the concentration of stresses and strains at that interface. And so the complexity of this enthesis increases during postnatal growth, which I'll talk about in a little bit. And this is in response to the loading environment. So I mentioned that tendon is a softer tissue compared to bone. Tendon is actually 2 times softer than bone. Bone is 2 times more stiff. And in the mature skeleton, and this can lead to a really abrupt accumulation of strains and stresses at that interface. And so we see not only is the tendon pulling on bone, but there's also shear and compressive forces that are acting at this interface. And that's kind of what drives the formation of this fibrocartilage. This tissue then is gradiently compositioned with a lot of small leucine-rich proteoglycans and elastic fibers incorporated into the tendon, whereas unmineralized and mineralized calcified fibrocartilage have compositional changes like aggrecan accumulation and collagen type 2. So I've been talking a little bit about bone and tendon, but the cellular and extracellular matrix gradients of the tendon-bone interface have both overlapping and divergent patterns compared to growth plates of long bones. And one area where these kind of overlap and diverge is comparing how growth factors like fibroblast growth factors are similar and or different. So one of the things that really caught our eye a few years ago were how and is reported in the literature as well, as the composition of the tendon-bone interface and how it relates to, looks like kind of like an arrested growth plate. So the tendon-bone interface often found at those eminences I was just referring to on the surfaces of bones. These eminences have hypertrophic chondrocytes that are similar to the growth plate. They have pre-hypertrophic chondrocytes and a proliferating cell population as well. Obviously, there's not cartilage on the above the proliferating chondrocytes. It's tendon instead, and these divisions are driven in part by signaling through FGF receptors, as well as with specific ligands such as FGF9. So we've been really focused on how FGF9, the ligand that's found primarily in the perichondrium, influences the growth of the tendon-bone interface, as well as some of our more recent work focused on the receptors like FGF receptor 1 and FGF receptor 3. And so you can see that they're spatially sort of separated. Sometimes there's overlap with these receptors and ligands, but these ligands signal through these receptors to initiate changes to the growth of the bone. So FGF9, fibroblast growth factor 9, is expressed in the perichondrium, like I showed in that schematic previously. And you can see this really nicely here on the left with in situ hybridization. So here we have a histological section of a mouse embryonic femur, and surrounding this is all the soft tissue like muscle and tendon. And you can see with an FGF9 in situ that it's highly expressed in the surrounding tissues and to a much lesser extent expressed in the bone. However, we know FGF9 is critical for bone growth because it regulates vascularization of the growth plate. And so in mice that have a global deletion of FGF9, we see impaired expression of CD31 and reduced vascularization of the bone in the embryo. We also see a delayed formation of the growth plate in the bone and reduced expression of vascular endothelial growth factor A. One of the other things that's noted in this paper from Irene Hung, who was a postdoc in David Ornitz's lab over a decade ago, is that the loss of FGF9 globally also leads to an increased size of the eminence or the deltoid tuberosity, where the deltoid tendon interfaces with the humerus. And we like to sort of screen through the literature every once in a while and try to find whole mount images of forelimbs that show the deltoid tuberosity, because we think of it as a really nice readout potentially of how muscle is loading the bone during growth. And so hopefully you can appreciate from this whole mount image here in the control forelimb, you can see the deltoid tuberosity is this shark fin-like shape here on the humerus. This tissue is, or this structure is much larger in the absence of FGF9. So when we globally knock out FGF9, this eminence gets bigger. What's interesting is they also found that the humerus and femur are shorter in the knockout mice. So not only is the eminence, the deltoid tuberosity larger, where tendon attaches to bone, but the bone is also shorter. So we kind of think of this as increasing the moment arm that the tendon can apply to articulate the shoulder. And so we wanted to dig into this a little bit deeper in the work we do in the lab. So as a postdoc, when I was at WashU, I started collaborating with Dave, Dr. Ornitz, and he was generous to provide me with several of his mouse lines that he had generated over the years. And I took those with me to my lab in Delaware, where I had a graduate student, Connor, who graduated over the summer, sort of tease out whether or not this phenotype of the deltoid tuberosity was real. And what he found was, yes, it is a real phenotype. We could reproduce it in our hands. Global knockouts of FGF9 did have enlarged tuberosities. This was not sex dependent. And we also found that these embryos, the deltoid tuberosities had a shift in how the chondrocytes hypertrophy. So they didn't necessarily have more hypertrophic chondrocytes in these tendon-bone attachments, but they had larger hypertrophic chondrocytes. And when we looked at these using histology with von Casa to stain the mineral, hopefully you can appreciate that they're these little brown bubble-like shapes are the where the mineral is being deposited, and there's a larger unmineralized region of the deltoid tuberosity and deltoid attachment in the FGF9 null mice, and there was also larger chondrocytes in these images. And so I mentioned that FGF9 is expressed in those surrounding tissues. These are some FGF9 lax-Z whole mount stains of embryonic day 14.5 limbs from mice, showing, you can see quite nicely the straight lines and the muscles that are expressing FGF9. We also looked early postnatally at the tendon attachment of the Achilles, and we found that the tendon highly expresses FGF9, and to a lesser extent, FGF9 is expressed in the bone. And so with higher amount of confidence, we support that FGF9 is derived from the soft tissue surrounding bone, and that's influencing the growth of this tuberosity and the growth of the bone. So then we looked at, we generated a transgenic mouse line that was specific to skeletal muscle. So we used floxed FGF9 mice and crossed them with an inducible skeletal muscle Cree. This Cree is ACT1, it's tetracycline inducible, and we were able to fortunately generate these mice and keep them alive postnatally. So one of the things I didn't mention, but is important for this study, is that we can't study the global knockouts of FGF9 postnatally because those mice die because their lungs are hypoplastic and they just aren't able to survive outside the womb. But these muscle knockouts of FGF9 are postnatal viable. They seem totally fine. And we're able to take those mice out several months and better look at the mineralization patterning of the deltoid tuberosity, as well as the 3D shape and structure of these bones. And so this work, also led by Connor and an undergraduate student, Jacqueline, who's now a veterinary student at Penn, did these studies when we were at Delaware. And so these are just some 3D reconstructions, microcomputed tomography or micro CT of these FGF9 loss of function bones when we knock out FGF9 in muscle. And hopefully you can appreciate from this 3D reconstruction and this 2D cut plane where this line is, that the deltoid tuberosity in young bones postnatally, the deltoid tuberosity is larger. We also took these mice out, like I mentioned, several months, and we looked at the 3D shape and volume of these attachments. And so Connor did a really nice job of segmenting these bones. Jacqueline did an amazing job of generating these mice. And we did find that the tuberosities in these attachments were quantitatively larger. So our hypothesis is that, again, we're testing that FGF9 is really important in these surrounding tissues, driving shape and morphology of the bone. And we have yet to, but part of our next steps are evaluating how FGF9 contributes to contractility of muscle and how that might be influencing the size and shape of the deltoid tuberosity. So I mentioned a lot of the work that we've done with the FGF9, the ligand side, but we also show that FGF receptors are important in the formation of these tuberosities as well. And so this was a study where we looked at postnatal knockout of FGF receptors using the COL1A2 inducible Cree. And so just briefly, this work led by Kendra Warnley, who is an undergraduate slash, when she graduated, she became a tech in the lab. She did a really nice set of experiments doing microcomputed tomography and histology of these mice that we received from the University of Chicago, where the COL1A2 Cree, when we give the mouse tamoxifen at 3 weeks of age, will recombine and label quite nicely with membrane tomato, membrane green. The green stain is the COL1A2 positive cells. And so you can see really nicely in this interface, this tuberosity surrounded by bone. The tuberosity is nicely labeled by COL1A2 positive cells, and that also labels the tendon. This definitely labels the diaphysis of the bone as well. But when we use this same Cree and dosing strategies with triple mutants, so FGF receptor 1, 2, and 3 are all floxed and crossed with the Col1A2 Cree, we see there's not a difference in the length of the bone in this triple knockout, but the tuberosity does get larger. And so this is when we knock out FGF receptor 3 postnatally, about 3 weeks of age, and take these mice out to 12 weeks of age. So as they grow, their bones lengthen, and that is not disrupted by this mutation, but the tuberosity size is. So that was pretty interesting to us, supporting that, okay, maybe the receptors are playing a role in the cells that are resident to the attachment and the tendon itself. This growth of the attachment was an interesting finding, and we're still trying to dig out what exactly the mechanism is. But a recent study in the Journal of Bone and Mineral Research by Jennifer McKenzie, led by David Ornitz, showed that loss of FGF receptors 1 and 2 in osteocytes in mice leads to, in osteoblasts and osteocytes in mice leads to death of osteocytes with age. And what they showed as well is that the knockout of these 2 receptors leads to an accumulation of bone mass, rapid accumulation of bone mass postnatally. And so these double knockouts, when they knocked out FGF receptors 1 and 2 in osteoblasts and osteocytes, you see this much thicker cortex, and then also the cells in the cortex die as early as 3 weeks of age, maybe earlier, and they continue to die, which may be contributing to the accumulation of this high bone mass phenotype. And so we wanted to see if that may be happening in some of our mice. We used a slightly different Cree, and this was a study that my research technician, Zach, did recently, where he looked at the attachments in mice that have the same 2 FGF receptors knocked out as this previous study, but instead of knocking them out in osteoblasts and osteocytes, knocking them out in tendon progenitors and interface cells. So using the scleraxis Cree, we looked at ligament and tendon attachments in the knee, and in the wild type attachments like the cruciate ligaments, you don't see a lot of tunnel positive stains, but in double knockout mice, so when we knock out FGF receptor 1 and 2 in the scleraxis Cree, you do see a lot of dead cells in the mineralized region of the enthesis, the tendon-bone or ligament-bone interface here. And so we think that FGF receptors 1 and 2 are required for cell survival in the mineralized region of this enthesis. And we're going to test this as well with FGF receptor 3 mutations in the near future on the RO1. So far, our data support that FGF signaling is important in interface growth. And I started this talk out by talking about muscle loading, and so I want to get back to that because that's really what's driving a lot of what we're interested in the lab. And so we're focusing, this next part of my talk will be focused on the work that we're doing controlling muscle contraction in vivo during growth. And that whole idea here is to modulate how the tendon is being loaded and then loading the interface to change the shape and structure of the bone. So interface development is dependent on muscle loading. I mentioned loss of muscle contractions can lead to reduction in the size of eminences. This is seen quite well with splotch delayed mice. These mice form muscles, but these mice do not form muscles. And their tendon interface, the eminence starts to form in the absence of muscle contractions. But as the embryo continues to grow, these deltoid tuberosities are not maintained. So they fail to continue to grow. So we know from years of work in developmental biology that muscle contractions are really important here. And for us, we can't really study the structure and function of these attachments in the embryo as easily because they're really small and not very mineralized and hard to grab onto, but we can study them postnatally. And we know that this growth of the interface happens relatively quickly postnatally. With 1 week old mouse Achilles tendons, the enthesis has a fairly uniform structure from the cartilage template into tendon, but by 2 weeks, this gradient starts to form. 3 weeks, you start to see the tide mark being established and the tendon becoming more aligned and more matrix rich. And then by 1.5 months, it's pretty much mature and fully developed. Bone is mineralized and growth plates aren't quite fused, but are starting to separate from the secondary ossification center. So we wanted to, so if we go back a sec, one of the things we are doing in the lab right now is diving in a little bit deeper as to what is the mechanobiological response of the tendon enthesis during this short time span of growth postnatally. And so we're using a technique called Optogenetics, which many of you have likely heard of. And it's a non-invasive tool to control activatable cells with light. And it's used in clinical trials currently for treating retinopathies because their photosensitivity, which is sort of displayed here. But it's derived from algae. So it's a typically a sort of light sensor in cells that's gives algae a direction to swim to the top of the water and undergo photosynthesis. So how does optogenetics work? Well, ions, inactivatable cells, ions diffuse through a channel and flow down a concentration gradient. And if the membrane potential changes enough, you can depolarize the cell and elicit an action potential. And in optogenetics, you can control the opening or the closing of these ion channels using blue light. And in our case, we're using a channel rhodopsin 2, which is responsive to 455 nanometer wavelength light. And that causes the ion channels in a non-specific way, cation channels, to remain open while the blue light is on. And so any cell that's exposed to blue light that's expressing channel rhodopsin on its membrane will keep its ion channels open and then lead to depolarization of the cells. And in an activatable cell, an action potential will be elicited. And so we didn't develop this tool for sure. This is like probably going to win a Nobel Prize eventually, but it is definitely something that's used heavily in neuroscience and has been used to control nerves for a while. And some studies have used it to focus on muscle contractility, but we're using it as a way of controlling muscle contraction in vivo to load the tendon and the bone. So optogenetics can elicit, I mentioned, both excitatory and inhibitory activity. These bacterial opsins, they're retinal binding proteins, and they, like I mentioned, combine a light-sensitive domain with an ion channel pump. So that light-dependent ion transport changes the membrane potential and then can, in bacteria, change its sensory functions. So these optogenetic tools are widely available. There's many to choose from on AdGene if you want to make your own AAVs, but you can also buy some of these mice through Jackson Lab. And these optogenetics tools can be channel specific, so you can tether them to specific ion channels, or they can be non-selective. Like I mentioned, we use channel rhodopsin 2, which is non-discriminatory against any cation channel. These optogenetic tools are and can be used widely in vivo, and we use them with Cre-recombinase models, but I mentioned AAV constructs can be used if you wanted to tailor them to more complicated mouse genetics as well. So first, we wanted to make sure that we were expressing these channel rhodopsins in skeletal muscle using the same ACTA1 CRE I was referring to earlier with the FGF9 study. And so we crossed an AI32 mouse from JAX with the ACTA1 CRE mouse and gave it doxycycline during gestation. And we see that there's a lot of channel rhodopsin 2 YFP expression, because this channel rhodopsin 2 is also bound to a YFP reporter. There's a lot of it in skeletal muscle, in the early postnatal mouse limb, but fortunately we don't see it in nerves, and we also don't see it in tendon. So we're pretty specific to skeletal muscle, which helps us know we're stimulating the fibers themselves and not eliciting contractions through the motor neuron. This will be useful for us in the future if we study age-related neuropathy or denervation, so we can directly stim the muscles with light. So Elahe, who is a recent, or Dr. Kanchi, who's a recent PhD graduate from the lab, focused a lot of her PhD work on this project. And her ultimate question, as I mentioned, was to exercise these, develop a tool to exercise muscles so that we could build stronger tendons and attachments using blue light. And so for her project, she first set to optimizing this model because we were the first to use it with direct skeletal muscle contraction to load tendon. We wanted to make sure that the loading protocol that we were using was going to maintain or sustain skeletal muscle contraction over time. So you can imagine this could be fairly arduous, especially if you have to, isolate or dissect the muscle or implant an electrode or something like that. But for us, it was actually quite straightforward. The hardest thing was anesthetizing the mice, which isn't that challenging, but we were able to elicit contractions on these mice without opening the skin or anything like that. There was no surgical intervention, and we could just remove the fur off the hind shank above the triceps muscle, and have a powerful enough blue LED that could penetrate through the skin. And so Elahe did a nice set of experiments trying out transdermal optogenetic stimulation of the triceps muscle, where she anesthetized the mice and used an Aurora in vivo ankle torque system to measure ankle torque as a function of light on-off cycling. And so she started with two different sort of frequency protocols, where she referred to one as 10 hertz, which was basically 70 milliseconds of the light on, followed by 30 milliseconds off, or a faster cycling at 40 hertz, which was 20 milliseconds on and 5 milliseconds off. And she could do these light stimulations using a LabVIEW controller, and eventually took these out for 20 minutes at a time. But at first, she just wanted to know, okay, can we sustain ankle torque over time with different frequencies? And what she showed was that keeping the light on for longer and allowing it to recover for longer elicited higher ankle torque over time. So we were sustaining ankle torque on average over time. Whereas if we use the if we use the faster cycling, we kind of exhaust the muscle a little bit, and we aren't able to sustain that load. And so moving forward, she used the 10 hertz frequency, and she compared different mouse ankle torque of different mice at different ages. And as one would expect with the young mice, their muscles are smaller, they're going to generate less torque than a larger mouse with larger muscles. And in fact, that's what she found. So she stimulated for 1 second intervals, 1 second over time of on-off cycles, and measured ankle max ankle torque of these and found that they trended quite nicely, correlated quite nicely with age. She then compared these to electrical stimulation, which is the gold standard, and she developed an electrical stim protocol that was a similar frequency profile as what she had for optogenetics. So we're not necessarily pushing these muscles to tetany, but we're matching sort of the contractility at the electricals through the motor neurons instead of directly through the muscle. And she found that ankle torque was comparable between the two. And then if she repeated this in the same mouse 3 days in a row on a cohort of mice, there was maybe a little bit of a drop of max ankle torque relative to the body mass of the mouse. Some stayed the same between electrical and optogenetic stimulation, and some declined. So we evaluated this as pretty comparable. It's not perfect, but it works quite well compared to the gold standard. And importantly for us, it's non-invasive. So then Elahe did a set of experiments to sort of test how well the optogenetic system can work to elicit changes in tendon structure and function, or in enthesis structure and function. And so she used this same platform and isometric muscle loading. So she fixed the ankle in position. So when we shine blue light on the muscle, the ankle doesn't move with the following protocol. So she used 5 days in a row of stimulation for 20 minutes per day. And then she let herself and the mice rest for 2 days and did it again the next week for up to 3 weeks duration protocol. She started this experiment in mice that were 3 to 4 weeks old, as well as another set of mice that we call the adult mice that were about 6 months of age. At the end of this experiment, she used a set of mice for micro-CT and histology, as well as for biomechanical testing of the tendon-bone interface mechanics. We also did a set of experiments looking at shorter time points, so after 1 week or 2 weeks of this experimental protocol, to do some RNA sequencing on tendon, as well as the calcaneus, as well as the skeletal muscle of the triceps surae. I'm not going to talk about that, but that's future work that we're going to hopefully present at ORS and in the future led by Saeed Alamia, who's a current grad student in the lab. Okay, so ultimately our question is, how does this interface adapt to mechanical loading, and how might it change between the immature enthesis and the mature enthesis? And so we looked at, as I mentioned, these two groups. Between loading, we used the contralateral limb. So we loaded the one limb with the optogenetic stimulation, and the contralateral limb on that same mouse is our internal control. So we can always compare back to that. And then we had another set of mice that were, I mentioned, that were older that we can compare back to see if there's an age-dependent effect. And so repeated loading in our study showed a distinct growth and opening of the growth plate, or maybe failure to fuse the growth plate. But this posterior calcaneus had a little bit of a bump out on the distal side of the foot, and the growth plate was wider in the loaded compared to the contralateral control. In the adult, we didn't see much difference, maybe a little bit of bone loss in the posterior side of the calcaneus, but not as much growth at that interface, maybe a little bit, but it wasn't as profound as the young. In the young attachment of the immature interface, we also saw this bump out a little bit more distinctly in histology, as well as increased prevalence of vascularization, potential vascularization at the interface. And as I mentioned in the beginning, the enthesis is not well vascularized. And so this was kind of a surprise to us that maybe this loading was inducing intravascularization near that fibrocartilage. We also saw more bone loss in the loaded side compared to the contralateral control. And what that led to for biomechanical properties was using uniaxial tensile tests of the tendon with it still attached to the calcaneus. We measure load and deformation, and we can calculate stress and strain from that. And we found that while the size of the tendon didn't change with loading, the ability of the tendon to carry load, the enthesis to carry load, was decreased in the young attachments. So they were mechanically weaker and less tough compared to the contralateral normally growing interfaces. And in the adult, we saw some potential differences, although our sample size was quite low. So we need to continue to do some of this work. But what we found was that maybe there was a little bit of an increase in the stiffness of the tendon and attachment with loading compared to control, which is different than what we see. We don't really see that trend in the young, and not too much of a difference in the load, although maybe a shift upward. 3 out of the 4 show that. So again, we need more data here to support this better, but it's potential that there's like a divergent effect of the loading in young compared to mature entheses. So far we've shown that optogenetics is this really nice tool to non-invasively and repeatedly model loading-induced adaptation in young attachments and adult Achilles entheses. And these findings model the structural changes that are similar to what we see in overuse activities in kids, especially young athletes. And we're digging into the mechanism a little bit more here, but we're interested in how this tissue might be accumulating or recovering from damage. And so one of the ways we're exploring this is using is looking a little bit into how collagen is remodeling. And so I mentioned that collagen is one of the primary constituents of this ECM of the tendon enthesis. And this collagen forms cross-links that help sustain its stability. Collagen denaturation is a marker of tendon damage. This has been shown quite nicely with Michael Yu's group at the University of Utah, where they've developed this detection method to mark collagen denaturation using collagen hybridizing peptides. And so when you accumulate mechanical damage, there's this non-recoverable damage that initiates collagen denaturation. And we can use collagen hybridizing peptides to bind to these sites of collagen damage that then fluoresce, and we can visualize how collagen damage is accumulating. So we've used these CHPs in vivo, and we've been able to track over time how these collagen hybridizing peptides accumulate. And in a really nice study we did at the sort of beginning of one of these experiments, where we did 1 week of 20 minutes per day stimulation, at the end of that week, looking at collagen damage using these CHPs, we were able to visualize using IVUS how sort of intense that CHP binding would be. And so this was a pretty neat study where we just directly injected the CHP from 3 helix into, subcutaneously near the tendon. So we have the heel here with the bone here, and then the tendon and the muscle up here. And this is just the mouse anesthetized and visualized under an IVUS system. And you can see, hopefully you can appreciate that the intensity of CHP binding is much higher in the loaded side compared to the control. We went on to look at these with histology, but unfortunately the histological process that we used ended up denaturing the denaturation tag. So we weren't able to look at them that well, but we're gonna do this again with some post histology methods to look at it more microscopically. So hopefully today I've convinced you that the developing enthesis relies on FGF signaling. It functions as an arrested growth plate, and FGF signaling regulates the size and shape of the bone ridges where tendons attach. Loss of fibroblast growth factor 9 in skeletal muscle can lead to increased enthesis and tuberosity size. And we've developed Optogenetics tools to non-invasively control skeletal muscle contraction in vivo, and shown that this tool can be used to increase mechanical loading at the enthesis and induce collagen level damage. So I'm not going to talk at all about the work that we're doing now, but focused on other areas, which include vasculature, hypoxia, and metabolism. But we definitely have a lot of this FGF signaling and mechanical loading work still going. And one of the other things that I think is kind of overlapping and really nice about the enthesis and the growth plate is that they're both hypoxic during growth. And so maybe someday in the future, I can come back and tell you more about that work, but that's a totally different area of research for the lab right now, but also an interesting perspective of like how these tissues form. One last plug I forgot to put in the beginning, but at the ORS this year, we'll have a workshop on career development for the trainees in the room. This is on strategies for negotiating across the career spectrum. And it will have 4 senior women faculty and industry representatives to talk about negotiating. And that's all. I wanted to make sure I make a plug for the wonderful people who helped with this work, like Olahe and Connor, Jacqueline, and Kendra, all did amazing work on these projects. So happy to take questions. And thank you for posting the link again. Appreciate that. Hi.
Thanks a lot, Megan. And just a reminder, everyone, please fill in the Google form that Dr. Killian shared. And we're open for questions now.
Yep.
You can type in your questions, or you can just speak up your questions, whichever way you prefer.
I raised my hand. Is it an order, or I can just go?
Yes.
You can go in and go ahead. It's okay.
Thank you very much. Extremely nice presentation, and I really like towards the end the way that you were able to visualize the collagen structure, and this is a thing that I remember that I really work hard to understand. And I remember Dr. Mwale kind of gave me like 3 books and like a year in explaining me how it works, so I can visualize something that now can be actually done in a much better way. So that was like, whoa! I will email you about that one specifically, out of curiosity. I have 2 questions, and I want to say I like your last cartoon, which it's so true and so much applies to me. And yes, I need help with the muscle agony. The tendon-bone interventions are extremely frequent because sport accidents happen, and they are kind of high profile at the same time. And the enthesis usually goes in 2 ways: the way that they're approaching it from a surgical point of view, direct and indirect. And I would like to know if there is anything that you would be able to recommend based on your research. What would be a better way to approach it to guarantee physiological reconstruction of the subsequent anatomical layers? And my second question is, and it's something that kind of like struck me very interesting, because you're trying to use blue light in optogenetics, and the experiments were done with blue light, at the and it's kind of like real blue light. It's around 430 nanometers, and and that blue light, it doesn't have a lot of penetration, and I'm curious, have you and it doesn't really carry a lot of energy because of the frequency and the penetration. So I'm curious, have you tried going low on the wavelength in order to increase the energy that you deliver, basically going into the violet light, and then going into the uh, deep ultraviolet, and I'm not saying you should go into X-ray and then go into gamma, because that'll be a little bit maybe too aggressive, but
Yeah.
kind of go into a little bit deeper into the near ultraviolet and far ultraviolet, because the increase of energy doubles in near ultraviolet and triples in far ultraviolet. And another thing that I think might work, and those are just I'm throwing a ball in here. What if you use two different sources of electromagnetic radiation, visible or not visible at this point, that actually they'll be able to be kind of uh, you that you can achieve a summation by basically being the double of the frequency, or every third frequency will accumulate, and you will have bursts of energy, because I noticed you're doing bursts of time, but I think you can achieve those bursts of time just by playing with two sources of energy, let's say with one in blue and one in violet, or one in violet and one in far ultraviolet, and then you will have you can go 10 times the energy without really doing nothing, and I think you will have a better penetration at the same time. And combining these two, why not applying to the tendon-bone interfacing reconstruction? Thank you. I know it was a very long thing, but I'm really interested in your views on this one.
Thank you. Yeah, so the great set of questions. So I'll start with the last one. So actually, I think I need to chat more with some of the boneheads in the group here at Michigan who study more osteocyte biology, or others, people in your group as well. But that is our osteocytes activatable. So we use the blue light specifically because it elicits depolarization in cells, and we're using it to control muscle contraction as an activatable cell. It is it's probably going to open and close ion channels in other types of cells, but whether or not it leads to something physiological has yet to be determined in non-activatable cells. But I think that that's an interesting question. The penetration of light. So it is important to use the specific wavelengths. We use 455 nanometers because of the specific channel rhodopsin that we're using. And there are other types of rhodopsins or opsins that can be used at different wavelengths. So there are some that work better with like far red activation. However, the band is pretty tight, and whether or not the cell will open or close its ion channels is very specific to that wavelength. We have done some really nice, Elahi did some really nice experiments showing how much the light refracts as it goes through the tissue, and how deep we can penetrate. We're still working on some stuff there, but I can tell you that the light is getting in. We know because it's eliciting a contraction. We don't know how deep into the muscle it's going, if it's hitting the soleus, for example, or other muscles around. But it's a really strong LED. We're at 1,000 megawatt LED. So it's it's very powerful. And so we're confident that we're doing something. But actually Saida's thesis is going to be focused more on what muscles exactly are we recruiting when we shine light through the skin or through these tissues. And then to answer your first question, the type of enthesis and how we repair it and what we need to do when we repair it is like such an important question. And I think we still don't know. And I don't think that there's any one great strategy yet that works for enthesis repair. It depends on the site and it depends on the tear type and the disease progression. Kathy Derwin has done some great work making rotator cuff materials to, help with overcoming the mechanical challenge of reattaching tendon to bone. Because one of the things I didn't talk about, but is really important, for example, for rotator cuff, is that when there's a tear in the tendon, the enthesis degenerates. We don't know if that happens first and is initiating the tear or what's going on, but we know that when the tendon starts to pull away from the bone and even is completely detached from the bone, it's really hard to pull it back to the bone and reattach it. And it's part of that is because the muscle remodels and shortens, and the tendon slips further away from its bony footprint. And so when the surgeon goes in and pulls it back to the repair or the original attachment site, it's under so much more tension than it was before it failed. And that also could lead to it failing again. But as far as like, how do we design these materials to help improve repair or scaffolds or hydrogels or whatever it might be, I think it is going to be driven in part by what's the state of the disease at the time. Like, what are we trying to stimulate? Are there cells there that can even do anything with that scaffold or that matrix or those growth factors that we put in, or those cells that we put in? And then do we just want to overcome that mechanical disadvantage of the detached tendon, or do we want to actually regenerate that gradient enthesis? I don't know that we necessarily, I probably shouldn't say this because that's part of the research that we're doing, but I don't know if we necessarily need to recreate the entire gradient of the enthesis, but certainly could see that being a beneficial thing to, it could help with mitigating this concentrated stresses at that interface. Does that answer your questions?
Oh yes, of
Great.
course, and I understand the wall that you have around this, because it's really worth making it work, basically. Thank you very much.
Yes, thank you. Gaiji.
Hi, thank you for the great presentation. I have two questions. My first question is, we saw there's negative impact of loading in young mice as compared to adult mice. I don't completely understand why is that so, because mechanical loading is important for the development of the interface, and in the young mice it is developing, and in the adult it is already developed. So why do we have negative impact of loading?
Yeah, I think maybe it's too much. In the young mice, maybe we're overusing the attachments or the tendons or the muscles a little bit. I think the mechanism as to why that's happening, we're actively looking into that, but I'm also not sure like what changes, like you mentioned, between the young and adult in the tissue structure, the ability for the tissue to kind of like respond. We're talking about a much softer interface in the young attachment versus the adult, and so maybe the adult one's just able to withstand those loads a little bit better. And one of the things, Elahe wanted to do so many things for her PhD, and I was like, fine, you just need to graduate and move on to the next step of your career. But one of the things she wanted to do was try like a training protocol before launching into the 20 minutes a day, especially for the young mice. Maybe it's just too much too soon and it's causing damage that's not recoverable. So, that's definitely something that we're planning to do with we people in the bone world that do anabolic bone loading do similar types of things, but we haven't done that yet.
Maybe I missed this part, but how young is the young mice? Like, is it, as you said, the interface is developed within like 1.5 months? So is it still in the development phase, or it is like already developed in the young mice?
Yeah, in the young mice, we're starting around 3 to 4 weeks. So it's not, the gradient has started to form, but the apophysis is still cartilage. We're not, the secondary ossification center is not fully formed yet in that calcaneus. And things are still pretty soft, we think. We really want to move earlier. And so we're working, collaborating with Ellie Zelzer at the Weizmann Institute to move it even earlier into the embryo, to study there where it's like, we know that there's no mineral gradient at that point. But for now, we were like, we can manage handling mice that are 3 to 4 weeks old. They're large enough that we felt comfortable putting them in the fixtures and things. If we move to younger mice, we will be developing 3D printed fixtures instead of using the Aurora knee clamp and stuff like that to hold things into place, because things are just so much softer.
I was just wondering if you say that it is the load might be too much for the young mice, it might be disrupting the matrix and everything. So do you think if we put less load, so it might be even beneficial instead of having negative impact, it could have positive impact on the development of the interface? Do you think that?
Yeah, I guess we have to define what beneficial is. And in this case, when we do our mechanical tests, we're saying it's like causing damage and it's weaker attachments, but we don't know if like over the lifespan, like if we were to let that mouse grow to be 6 months of age, would its attachment be stronger by then? So we don't know that part, but I think certainly like, so that was like a side blurb, but the beneficial effects are kind of challenging to define for the growing skeleton, I think. And so I think for us, we would say, okay, are we, is it growing in size? Are we, getting the tissue what we want it to look like? And I think we aren't, other than like maybe we're increasing vasculature a little bit and we're decreasing the mechanical strength, but I don't know if we know if it's like over the lifespan a bad thing, what we're seeing. So I want to temper my response a little bit in a non-answer for that, because I don't know if I do think that if we did a training effect and we like introduced them slowly to the exercise, we would see maybe more anabolic response. But then you also get some things with like you might not reach the set point of being able to be above a certain threshold that it would induce a change. So I think if we focus a little bit more too on what's happening at the cell level, what are the cells seeing and sensing and responding to? How are they, depositing matrix or repairing damage at that interface? That would be a really interesting next step for this project too. So I didn't really answer your question because I don't know.
It's good knowing that. Can I ask one last question, please? We saw that when we knocked out FGF1 and 2, we saw loss of cells in tendon-bone interface. Do you think we have similar effects in the tendon when we knock out FGF1 and 2?
I don't think so. So those data were, I don't know if I can go back to that slide, but yes. So the slide, so this study, we are knocking out FGF receptor 1 and 2 in tendon as well. And we don't see the cell death, at least at this time point, which was 3 weeks old, that we see in the bony region of the enthesis. So I don't know if FGF receptor 1 or 2 are like contributing a lot to tendon. You know, one thing might not be efficiently recombining it in tendon or something like that, but we're gonna. I was surprised by this, and I was surprised to see that maybe tendon isn't as affected by the receptors as the enthesis is. So, to answer that question.
Yeah, thank you so much.
You're welcome.
So thank you, Megan, for such a wonderful presentation. I like the whole area of mechanobiology, and especially as it relates to the enthesis, tendon healing. Really wonderful. So actually, my question is related to the questions that Gagi was asking you, so it's when it comes to loading, there's a sweet zone or a Goldilocks zone, so to say. So what do you think is the sweet spot for the rotator cuff healing, and is it moving with age? Because if you like you said, if you overload, you're in trouble. If you underload, you're in trouble. So how does that move in your?
Yeah, that's a really good question. I don't know. So, and it also depends on disease state, right? So a healthy rotator cuff, you want to probably have a very different set point than if it's been torn or has been surgically repaired. So following injury, for sure, you need to allow the tissue to heal. And there's been some really great studies that have explored that, sort of complete loss of loading with denervation of the muscle and immobilization with casting. That's not good. You shouldn't do that. You should allow some range of motion or prevent some scar formation from happening. But if you go too soon, too fast, too high of load into reloading that tendon after it's been repaired, then it will fail, or it could fail, right? You have to let some of the residual or the healing tissue set up. And so that healing process, what that scar is experiencing, how loading on that scar or that fibrous tissue between the inner tendon and the bone is remodeling is, I think, a fairly open question. Other areas of research are exploring that, but for the rotator cuff, I'm not super familiar with what that would look like and how you could translate that to an in vivo system, right? So I think we could start with experiments with cells in a dish, a matrix in a dish, and look at it that way. But how does that translate in vivo? It's a really interesting question.
Right. So yeah, thanks. So for your optogenetic activation experiments, I really like the methods. It's really elegant and really beautiful. Really want. So for again, I'm coming to this optimal thing. How do you arrive at your optimal pulse optogenic stimulation? Do you have, when you're starting, you have maybe a couple of hertz, 10 hertz, or do you just, how do you get to that?
How did we get to 10 hertz?
10 hertz.
Yeah. So it was based a bit on some data, previously published work by, there's a paper in Nature Communications. I think the first author is Brugman. They did a set of experiments with isolated muscle and looking at sort of on-off cycles and max force generation there. So we started there with the in vivo setup. Elahe did a set of experiments, sort of trying out a couple things. She tried out changing the off time, changing the on time, but with a fairly, like, I guess the readout was the ankle torque, right? So it was repeatable, but definitely, like, you have to like let the mouse rest, and you want to test the same mouse a couple times. So it took some time. So she sort of started with a set of just single pulses to see how much torque she could generate without doing the repeated torque. And from that, just narrowed it down. Okay, to generate the max force or the max torque, we need the light on for a single pulse for 70 milliseconds. And so from that set of experiments, then she went to the cycling. So the first one was just single pulses and letting the mouse kind of re-equilibrate, the muscle re-equilibrate before doing the next ones. Similar to how we would do like muscle mechanics with contractility with electrical stimulation or nerve stimulation. So following those sort of established methods in electrophysiology, but we're using light pulsing instead. And one important thing, I think, it can vary depending on which channel or which opsin you're using, of how fast that membrane potential can reset. So some channel rhodopsins or some opsins have a slower decay, and some have really fast decay to re-equilibrate. And so that was like, we just kind of got lucky with the channel rhodopsin that we're using is designed to be a faster reset. And so we didn't try different strains, although we definitely have like, we want to try the inhibition side of things. But yeah, I think that, does that answer that your question there?
That's really brilliant. Thank you very much.
Thank you.
Any next question? Anyone? So Megan, I have a question for you.
Sure.
How do you think the enthesis is formed in the spinal column? Like the loading is entirely different.
Yeah.
And even in gestational and non-gestational phase or like postnatal period, but like if you look at the spinous processes and the transverse processes, they're already there. They're part of the pull force from the muscle of the back. So how are they forming? Like, any thoughts on that?
Yeah, so like the spinal processes, I think I actually don't know too much about. Some of them are fibrocartilage and some of them are fibrous. Is that right?
All these spinal processes like lateral and the transverse and the other ones are all bony, and they are there when we are born. So I don't know what process makes them while we are in utero, like we're floating or we're floating. And then, so where is the load coming from? I mean, the child or the fetus moves for sure, but is it enough pull on the back in the wiggly space to pull and create that enthesis? Or that?
And it probably depends on where along the spine you are, right? So you might have in the spine, there's shorter processes depending on, and longer processes depending on where you're at. And so I think it's a similar patterning. The timing might be different, but I think like the structural organization of the interface. I mean, so I should say, depending on like there's a really nice study from Ellie Selzer's group, led by Netta Felsenthal, showing that there's a difference between stationary and migratory attachments, right? And so the spine probably has stationary attachments. The cell patterning in those attachments is probably different, and they're still experiencing mechanical load from the spinal muscles. And they probably don't have, depending on where you're at along the spine, perfectly normal forces. Like what I mean by normal, like just perpendicular to the insertion, right, or parallel to the muscle plane. So there's some movement of that spine back and forth that could lead to fibrocartilage-like enthesis. So I think it depends on how much mobility in the spine there might be and how much motion that it's experiencing. But certainly, like, there was a really nice paper from Neve Nolan, and she does a nice set of experiments looking at muscle unloading and skeletal growth. And I think she had it with one with spine recently in JOR that might lead to that. But that's a huge area of
Yeah.
I don't know much about spinal entheses. I can't imagine that they're that much different than the particular entheses, but the muscle loads are very different, certainly, yeah. But
So
enthesis to enthesis are so different, and tendon to tendon,
exactly.
they're so different. So, yeah.
Exactly. So in your mouse model, maybe it's worthwhile looking at the CT scans of the spine to see that whether there is a difference between these spinal protuberances, like if FGF plays a role. I don't know.
Yeah,
maybe.
That's a great idea. Yeah.
So the other question I have is about the ion channels. I mean, I was just thinking that instead of the UV or the whole tanning beds, if they had the blue lights in there or had alternate bulbs, that might be more useful. But so which ion channels are primarily activated, do you think? And because it's not activating your nerve or the nerve.
It's not activating through the motor neuron. Yeah, correct. It's not nerve stim with this particular setup that we have. Previous studies, I mentioned the Brugman paper from Nature Communications in 2016, I think, and there's a McGill paper from similar time frame. They used Crees that targeted muscle, but also targeted nerves. But in one of those studies, they showed it's primarily driven through sodium channels, but it also it activates. It's non-specific to cation channels, so it will be opening and closing, or it will be opening calcium channels as well. So that is, we did a brief study looking at calcium signaling with the optogenetics, and we found that optogenetic stimulation does lead to a shift in how calcium signaling is affecting muscle in the short term, and some of the RNA-seq data support that as well for the longer term loading. But we also see like inflammatory response to the optogenetics, and that could be driven because we're basically hammering these muscles and skin with UV light, right?
Yeah, because that's what I was thinking, that it should ideally not be through calcium, because that would cause local spasticity. I mean, but which is good that it can be in a spasmodic phase and just keep on pulling on the disc or on the protuberance. But at the same time, it's not good for the neuromuscular junction because they go into refractory phase.
Yeah,
unless it's flaccidity.
Yeah, it's interesting. I guess we could potentially look at where the neuromuscular junctions are and see if it overlaps with some of the channel rhodopsins receptor, like on the membrane too, to see like are they overlapping or not. But yeah, you can. I think there are some sodium specific opsin targeting models that could be used. It's a really good point.
Yeah, thank you. So is there any other question? So please fill in the Google form shared by Dr. Killian for sure.
Thank you.
I filled in mine. It was an amazing talk. I mean, it's just amazing and very good body of work. And I look forward to talking more about this at ORS. And on behalf of the program and the program director, Dr. Mawali, we'd really like to thank you for enlightening us and giving us this beautiful lecture this afternoon.
Well, thanks so much for the invitation. It's great seeing you. This is by far the most people I've seen on a Zoom in a while, so I really appreciate you all attending. It means a lot.
Soon we will be all in person and able to travel across borders. So
exactly.
Yeah. Thank you very much, Megan. Really wonderful talk. Really appreciate it. Hope to see you at the ORS.
See you all at ORS. Yeah.
Yeah.
Thank you.
Bye.